Hello everyone. InformationBoxTicket Lifestyles brings you today Mycology topic on Candida tropicalis. But before starting this video, don't forget to like this video, subscribe to my channel and press the bell icon button. Let's see the table of contents today. First, we'll learn what is Candida tropicalis, habitat of Candida tropicalis, morphological features, cultural characteristics, pathogenesis, clinical features of Candida tropicalis infection, laboratory diagnosis, treatment, Candida tropicalis, antifungal resistance, prevention and control. Let's begin with the introduction of Candida tropicalis. Candida tropicalis is a yeast type of fungus that is pathogenic in neutrophilic hosts and spreads to peripheral tissues via the bloodstream. In pathogenicity and clinical characteristics, it is very similar to Candida albicans. It is a prevalent systematic fungus that affects the people with compromised or suppressed immune systems. Amphotericin B, echinocandis, and broad-spectrum triazole antifungals are common treatment of Candida tropicalis invasive infection. It is also known to induce infections when the human host normal microbiota has been compromised by the use of antimicrobial agents such as antibiotics or when sugar levels in the systems are elevated. Now let's see the habitat of Candida tropicalis. Candida tropicalis is a fungal species found in salt water, sea sediments, mud flats, marine fish intestines, mangrove plants, and marine algae as well as shrimps. As a result, they are common in tropical or subtropical marine habitats. It is also found in high concentration in human gut, fruit surfaces, a range of foods, and soil. Morphological features of Candida tropicalis. Candida tropicalis is a round or oval-shaped vegetative cell with a width of 2 to 10 micrometer. These Candida fungus are known to create colored pigment on cultural media that are red, pink, or orange in color. They are dimorphic and give rise to single-cell blastocornadia. They multiply by unipolar or bipolar budding. The blastocornadia buds are produced on needle-shaped stalks. The buds are pigmented and septide, which form pseudohyphae. Cultural characteristics of Candida tropicalis. Candida tropicalis thrives at temperatures ranging from 25 to 35 degrees Celsius. Their growth flourishes in sugar and fat media. Abroad dextrose agar or yeast potato dextrose agar yields cream-colored or off-white to gray, bland, smooth, soft, and creamy or wrinkled or rough results. This medium inhibits conidia development while promoting mycelium formation. Conidia flourish on cornmeal agar, blastocornadia. They can also thrive in potato dextrose agar, potato glucose, potato carrot, tomato juice, and lima bean media. Now let's see the pathogenesis of Candida tropicalis. Candida tropicalis is one of the most prevalent Candida species that colonize humans, causing pathogenic infections on the skin, gastrointestinal system, and female genitourinary tract. Transmission of Candida tropicalis. Nosocomial infection occurs most frequently between healthcare employees and patients. The fungus, which is most commonly found in immunocompromised individuals with HIV, AIDS, cancer, leukemia, and organ transplantation, produces candidiasis in different organ systems of the body. Virulence factors of Candida tropicalis. Blastocornadial spores are immune to a variety of environmental variables, allowing them to flourish in both natural and human host settings. As a result, they can live on hands for more than 24 hours before being transferred to another host. Candida tropicalis colonization skills have been related to the development of a biofilm on the exterior of the epithelia and endothelia cell lining. Adherence is caused by the secretion of enzymatic enzymes such as protease, which enables the fungus to infiltrate the epithelial lining. Protease enzymes aid in the hydrolysis of peptide bonds, allowing bacteria to adhere and colonize deep tissues, resulting in organ damages. Phospholipids hydrolyze phospholipids, allowing them to pass through a hyphal end into the epithelial cell membrane and into the cytosol. Other variables that contribute to fungus colonization on epithelia and endothelial cell linings include immunocompromised system of the host, long-term use of antimicrobial drugs such as medicines, the use of tubes for an extended length of time, and lastly, antifungal overuse. Clinical features of Candida tropicalis infection. Candida tropicalis infections differ depending on where the fungus are found. Among the symptomatic symptoms are number 1. Superficial and mucosal candidiasis. It is linked to oral pharyngeal candidiasis, angular cheilitis, and vulval vaginal candidiasis. Oral thrust, also known as oral pharyngeal candidiasis, is the colonization of the tongue and the oral area by Candida albicans. Candida tropicalis colonizes by secreted enzymes that enable it to escape the host's immune system and colonize the epithelial lining. It can also induce superficial skin and nail diseases. Onychomycosis, diabetes, catheterization, and leukemia patients are all at risk for candidiuria. Candida tropicalis causes widespread superficial illnesses such as interdigital candidiasis and diaper rash. Number 2. Systematic candidiasis includes disseminated candidiasis, gastrointestinal candidiasis. Candida tropicalis colonizes the gastrointestinal system in individuals who are at high risk of developing invasive candidiasis. Candidemia. It is a growth and impact of Candida tropicalis on the human host internal systems. It is prevalent in cancer patients suffering from leukemia or neutropenia. This illness can be contracted in the hospital. Number 3. Chronic disseminated candidiasis. This is a serious case of candidiasis spread in leukemic and secondary neutropenic individuals. They have an impact on liver, pancreas, and kidney. Kindly show your support by subscribing to this channel. Laboratory diagnosis of Candida tropicalis. The specimen is skin scarring, sublingual and genital samples, tissue biopsies, blood, cerebral spinal fluid, or pee are all procedures that may be used. Next, microscopic examination. Pseudohypha detection using a 10% potassium hydroxide moist mount from skin scrapings or tissue samples. Differential stains such as hematoxylin and eosin, periodic acid Schiff, and Gomory, Corcot, methamine, silver stains are used in differentiate fungal infection in tissue slices obtained from biopsy or autobiopsy tissues. Cultural identification. To create smooth, shiny colonies with the development of small conidial spores held by a hyphal stalk structure, use sebroid, dextrose agar, and or yeast extract agar. Smooth Blastoconidia spores are grown on cornmeal agar. Next, biochemical characterization. The purpose of this test is to identify the generation of acetic acid, fermentation on different sugars and indocetol elements synthesized in carbon sources. Urease production is prevalent among Candida tropicalis families. Molecular detection. Polymerase chain reaction for the detection of genotyping. Let's head to the treatment of Candida tropicalis infections. Antifungal drugs are used to treat Candida tropicalis infestation. Amphotericin B is most frequently used to treat Candida infestations. Topical antifungals are antifungals that are taken directly or applied locally. Topical antifungals like nystatin are used to address superficial skin and nail diseases. Mouth antifungals are prescribed to address mouth candidiasis and gastric candidiasis. Flucytosine-based medications such as caspofungin, micafungin, and anidulafungin are used to treat oropharyngeal, esophagus, and invasive candidiasis. Voriconazole, a new generation broad-spectrum fluconazole, is used to treat recurrent candidiasis in HIV/AIDS patients who have accurate fluconazole tolerance. Candida tropicalis antifungal resistance. Fluconazole has acquired tolerance in Candida tropicalis over time, so it is no longer suggested for the treatment of recurrent candidiasis. Fluconazole inhibits the phospholipase connection to host epithelium cells, which aids in adhesion. If its mechanism of action is ineffective, itraconazole, voriconazole, phosphorconazole, rovaconazole, and isavuconazole are extremely active drugs. Voriconazole, a new generation of broad-spectrum antifungal fluconazole medications, was designed to combat Candida tropicalis resistance. And lastly, prevention and control of Candida tropicalis. Hand cleaning in order to remove fungal spores from the palm and avoid the spread of the fungus. Using of water to clean affected areas. And that's it for today. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel, and press the bell icon button so you don't miss any of my videos. Show your support by subscribing. Thank you.